Well, thanks. That's a great honor and pleasure to speak in this series. Let me just upload the talk. Right, frontal lobe function and the psychopathology of everyday life. So I should say that frontal lobes really pose one of the most severe scientific challenges for us in the future, understanding how they work. And they've always had rather mysterious aura about them, ever since really this classic case of a brain damaged subject who threatened to illuminate the functions of the frontal lobe in the 19th century. So this is the singular case of Phineas Gage. So Phineas was a foreman, a railwayman, working in Vermont in the States. And he was involved in dynamiting rocks in order to lay railroads. And one day, unfortunately for him, he blew a huge tamping iron, as you can see in this illustration, clean through his head, through an explosive. It entered in the left side, the lower jaw, and exited the skull, and took with it a huge amount of blood and brain. So he collapsed on that, unsurprisingly, and people assumed he was dead. But after several convulsions, he was still alive. He was taken back and nursed back to health over the next few days. And rather remarkably, it transpired within weeks that Phineas was almost back to normal. So his basic functions of talking and walking and showing cognitive functioning seemed to be intact. Gradually, it became clear that his personality had changed. And previously, he'd been a temperate man, his personality full of energy, so it was said. But following this damage to his brain, he became capricious, vacillated, indecisive, irritable, profane, irreverent, and emotional. So it was said. There was quite a lot of controversy about this. His case was almost resurrected in the last century by Tony Damasio, because the tamping iron and his skull were preserved, and Damasio and his colleagues were able to do some magnetic resonance imaging studies and reconstruct the course of the damage, if you like, or the course of the tamping iron through his skull. And here you see that trajectory, damaging not only the whole of the left frontal lobe, that part of the frontal lobe behind the eyes, which is called unimaginatively by anatomists as orbitofrontal cortex behind the eyes, but it also extended into the right hemisphere as well and damaged some of the right lobes. And as you'll see later in the talk, Damasio used Phineas Gage in order to produce his theory of frontal lobe function. Now, it's still somewhat controversial, the degree to which Phineas Gage showed changes in personality, because it wasn't recorded very well and people weren't that sure about his personality beforehand. But nevertheless, this is the dawn of the era of the mysterious frontal lobes. Now, a dozen years later or so, rather more specific findings occurred from the neurologist Paul Broca, working in France. And Paul Broca discovered a patient called Monsieur Tantan, who suffered a stroke, which it transpired later had damaged quite large parts of the left frontal lobe. Here's a picture of the brain, his actual brain, and the damage to a particular area in the left frontal lobe, now called Broca's area. And Tantan's problem is that he could understand language, but couldn't express it very well. That's a gross simplification of his problems, which were much broader than that. But this was some of the first evidence for localization of function, a specific function, in this case language, and the flexibility and fluency of language associated with frontal lobe function. Intriguingly, other quite famous individuals have suffered strokes to the same area. This gentleman you may recognize as Baudelaire, of Fleur du Mal fame. He suffered a stroke, and then from thence on was really reduced to saying the word crénon, crénon, crénon. It's the only word he could say. Somewhat profane word, but that's all he could repeat. So he could understand language, but he couldn't express it. And another famous character, Vladimir Lenin, as you see here, also had a stroke. He donated his brain to a psychopathological institute in Germany after death, and he was restricted to saying vol vol in response to almost any question, although again, it seemed as though he understood what was being said to him. So thence the frontal lobes implicated in a basic function of language, fluency, articulation, and in fact, now as we understand it, grammar and syntax, organization of your behavioral outflow. Now, looking at the frontal lobes, these are pictures of the frontal lobes, and you can see the well-known sulci and gyri of the brain. The sulci are the ravines, the gyri are the folds of the brain. This is the frontal lobes of the brain. The motor cortex occupies area 4 here, and other motor area 6, and the rest of it is called the prefrontal cortex. Again, one of these peculiarities of anatomy. It's really the frontal cortex, but it's called the prefrontal cortex. And you see these funny numbers on it, both from this left view and the medial view here. This is looking at it from the middle of the brain on one hemisphere. These numbers are called Brodmann areas, after the histologist who discovered that there are different types of cell, different types of nerve cell, histological changes within these areas which characterize them. And of course, that immediately raises the interesting question of, do these areas have different functions, and how do they all work together to produce frontal lobe function? Now, in terms of nerve cells, I'm aware that many of you probably are not neuroscientists, and so I should refer you to the great Nobel Prize winner, Romani Cajal, in the early part of the 20th century, and his so-called neuron doctrine. And using really amazing techniques for his time, he was able to stain neural tissue. It is actually a section through the frontal lobe, through its 6 layers, and you can see these nerve cells with their nuclei and these amazing branching axons and dendrites, which make connections with one another to produce brain function. And here you can see a beautiful neuron and cell body stained here, and the incredible branching of these neurons, of which there are about 10 to the 11 in the human brain, probably forming around 10 to the 14 connections. So the frontal lobe has some rather specialized cells as well, which is very interesting. It has some giant pyramidal cells called von Economo cells, these gentlemen here, which seem to be very important to it. Now, in modern days, we look at connectivity of the frontal lobes and other regions of the brain using brain scanners, brain imaging. And one thing one can do is to have someone lay down in a brain scanner and not think of anything in particular, but just measure the fluxes of what's called resting state activity in the brain, changes in blood flow, essentially in the brain, which approximate neural activity. And this is a representation taken from some work of Ed Bullmore and Paula Vertes in psychiatry, showing you a new mathematical way of understanding what's called connectomics in the brain. Now, this is the frontal lobes anatomically. Note in particular these yellow dots here. If you apply connectomics to actually look at what are the hubs and spokes of the brain, this is what it looks like. You can see this transformation. Wow, now look at that. Doesn't look anything like a brain, does it? But it's a small world network, a bit like airports around the world, which are optimized for communicating aeroplanes. This one has hubs in the frontal lobes around these yellow dots in particular. So you can see those here. So I repeat this. Here we go. And these hubs are very important servants of the frontal lobe. They have lots of connections from other parts of the brain, and they influence virtually all other regions of the brain at different levels. So that's the basis of what you call an executive system, something which reaches out not only to its peers, but also to its underlings, if you like, and makes contact with them and synthesizes and integrates that information. So the neurons themselves form synapses. So these are two stained neurons, and they use neurochemical transmission, which means that electrical impulses, when they pass down the neuron, don't pass directly to the next neuron, but there's a gap there which releases a chemical neurotransmitter, which causes ionic changes, which propagates the impulse into the next nerve. So that's how communication in the brain actually works. These chemical neurotransmitters, of which there are probably over 100, include the famous dopamine, which I'm sure you've all heard about. And dopamine is released in certain neurons following a neural impulse. And when it's released, it diffuses across the synapse or gap, occupies these receptor proteins on the postsynaptic neuron or nerve cell, and then propagates electrical impulse subsequently. So that's basically how nerve cells work. And all of them work more or less in that manner. Now, interestingly, drugs such as cocaine and amphetamine work by manipulating this process. They, for example, block the reuptake of dopamine back into the nerve cell and therefore increase the amount of dopamine in the synaptic gap. And drugs like cocaine, amphetamine, and Ritalin, also called methylphenidate, which is used to treat hyperactive children, no less, all work in much the same way, although with different time constants to boost dopamine transmission. So what I'm going to be talking to you about really is the intersection of these chemical neurotransmitters in the brain, which all arise in the brain stem and lower levels of the brain and rise to innervate the so-called cortex, including the frontal lobes, as you see here. So this is the dopamine system here. And it's well known now from experiments in experimental animals and humans that these systems are implicated in such functions as stress, arousal or alertness, mood, reward, and so on. And I'll illustrate how these interact with frontal lobe function as we go. Now, I mentioned executive function earlier. And how does one measure executive functions exactly in a neuropsychology lab? One way of doing this is to give people lots of different tests which have different components, different types of cognitive challenge. So one kind of cognitive challenge is working memory. And that's typically measured, or very simply measured, in the following way. Can you remember a phone number? For example, if I say 365878, can you all recite back to me 365878 very easily? That's a test of short-term memory, right? I'm sure you're practicing it now. But that's not quite working memory. Working memory is working on the memory, working on the input. So if I say to you, I want you to repeat my phone number backwards, 365878, then you're using working memory, okay? It's a way of converting perceptual input into representations in the brain which outlast the sensory input. And you work on these representations to produce thinking and other outputs. So working memory is one of these functions, but there are two other important ones in this triad of executive functions. One is inhibition, the ability to stop doing something or to inhibit an impulse. And another is shifting, to show cognitive flexibility, to move from thinking about one thing to another. If, for example, the environment changes and it's important to flexibly change your behavior. So you can have a whole variety of these tests and look at their intercorrelations. and do what's called a principal component analysis. And you find indeed that tests of response inhibition and working memory and shifting do crystallize out in that way. But they're all somewhat related to one another. And so we call the whole thing executive function. Intriguingly, working memory is the component which is most related, as you see here, with these very high correlation coefficients, to IQ. So it's often been said that the frontal lobes mediate IQ or intelligence. And there is some truth to that, but it's not the whole story. Working memory certainly predicts your IQ. So a working memory test can predict this quite well. But tests of inhibition and flexibility do a less good job, as you can see here. So there's something extra that the frontal lobe brings to the feast, if you like. So let's look at some of these components separately. So one working memory test I've already shown you is a verbal one. So reciting a telephone number backwards would be verbal working memory. And I could block that for you if you had to say at the same time as you were remembering my phone number backwards, the expression the. That would prevent you rehearsing subvocally my phone number, and you wouldn't be able to remember it. It interferes with working memory. Now, a rather different form of working memory is visuospatial working memory, as exemplified particularly, for example, in chess. This is a chess position. I don't know if I've got any chess enthusiasts listening in here, but this is a position which, you know, white can win in 3 or 4 moves. Can you see the solution? Well, I'll tell you briefly what it is. It involves cleverly a sacrifice of this white rook taking the pawn, removing the protection of this square. So when the black rook takes the rook, the queen can now come in here and check. The king has to go to this square, and then this rook swings into action here to deflect this black rook, which takes it, leaving this square naked to the queen's checkmate. A 4-move checkmate with 2 rook sacrifice. But you have to see ahead. You have to look ahead here and use working memory to manipulate these visuospatial patterns. Now, if you interfere with visuospatial working memory, you can do so very easily simply by tapping a sequence. That's enough to completely put off the very best chess players from solving this problem. What we do in the lab, we have worked with chess players and chess, but it's a difficult game to put into an experiment. And so we've used rather artificial problems called the Tower of London, or we call it the Stockings of Cambridge, because you have to imagine that this presentation here is if you like, snooker balls in pockets, and you have a lower arrangement and an upper arrangement. And I want you to tell me, reaching into these pockets, how many reaches and moves would it take to convert the bottom arrangement into the top or goal arrangement? Well, the answer is exactly 5 moves is the optimal solution. And this is rather chess-like because you have to visualize the sequence of moves that you do in your head here using working memory. And intriguingly, patients with damage to the area 9 and 46 in the frontal lobes are very bad at working memory problems of this type. And so this area of the frontal lobe is specialized for certainly spatial working memory, particularly on the right side of the brain, actually. Now, the other way of looking at this is to do brain scans where the subject works with the touch screen and does the problems while lying in the scanner here. And the activations you get in the brain, shown in yellow and orange here, this is the right frontal lobes. You can see this activation in the right superior frontal gyrus, dorsolateral prefrontal cortex. There's a big activation in the back of the brain because it's a visual test where visual function is mediated, and the frontal lobe is coordinating this visual network. And here's the activation on the left side of the brain. So these bits of evidence show that this type of function is mediated by one part of the frontal cortex. And we have other corroborative evidence from animal experiments, which I won't go into in detail, but I will say that just as the human brain has these very different areas, as you see here, so does the monkey. There's a lot of what we call homology, or conservation of frontal lobe areas across species. Even the rat and the mouse has some frontal lobe, not as well developed, of course, as humans, where it occupies almost a third of the entire cerebral mantle. Now, in the monkey frontal cortex, when they do a working memory task where they have to remember where the location of a rewarding peanut is after a delay, they find neurons in this area which seem to bridge the delay. They hold the information beyond the presentation of the cue until the response is required. So they're mediating this working memory function in all probability. The second of the functions I mentioned is cognitive flexibility. Cognitive flexibility is measured in a number of tests in the laboratory. For example, you could switch from two tasks where you have to name letters or name numbers as quickly as possible. And it takes you some time to change over, which you can measure very accurately. And that time is lengthened with frontal lobe damage in certain regions. And here's a couple of tests. I'll talk about this one later. This test can be given to humans and monkeys, actually. But it's based on the Wisconsin card sort test, where you have to learn a rule for sorting cards. Do you sort them by color, shape, or number? You get feedback from the clinical psychologist who says, correct, correct, correct. So you eventually learn, oh, color is the thing to sort by. But then suddenly the feedback changes, the rule changes. And patients with frontal lobe damage, as Brenda Milner, the famous neuropsychologist who was trained in Cambridge in the 40s and 50s, frontal lobe patients are very bad, not at learning the task, but at shifting the categories. They perseverate in the same rule, even though they know it's changed. There are many other ways of testing cognitive flexibility, but these are just a few. Now, you might ask to what extent this kind of abstract test has any reality in the real world. Well, one of my brilliant graduate students, now research fellow at Churchill's, Leo Zemigrod, did this study on the web with a very large number of volunteers, looking at the relationship between performance in tests of cognitive flexibility, including the card sort test, and Brexit attitudes. And she found a very strong relationship such that the poorer the performance on the test of cognitive flexibility here, the greater the positivity in attitude towards Brexit. And similar type of questions, for example, positivity towards immigration went in the opposite direction. Now, this wasn't simply an effect of intelligence. It wasn't the case that the Remainers, if you like, were more intelligent. They were actually just more flexible than the Brexiteers, according to this survey. So that shows you how something like cognitive flexibility, abstractly defined by these tests, frontal lobe tests in the laboratory, actually can be related to attitudes in the real world, which is part of the theme of my talk. Now, I was introduced by saying I do a lot of work on disorders, and certainly it's true to say that virtually all of the major mental health disorders have dysfunction of the frontal lobes in some way or other. I can say that almost unequivocally. Schizophrenia, depression, anxiety, addiction, obsessive compulsive disorder, which of course is a very, very disabling disorder, very severe. This is hoarding. I often say in my lectures that this is a typical office of a Cambridge University Don, which can bring a laugh, but sometimes doesn't. OCD can involve barbering, just simply pulling the hair and causing baldness. Hand washing, contamination is a very likely one, probably exacerbated in the present dilemma we're in. Perfectionism comes into OCD. And obviously fairly famous individuals have been said to have OCD, including Beck's here, and a few others. Martin Luther, very strong, almost clinical OCD when one reads details of his biography. Samuel Johnson. Charles Dickens had mild OCD. Obviously, this didn't impair some of their major achievements. Hans Christian Andersen. And of course, you probably all know about Howard Hughes, contamination fears. Well, we've studied cognitive flexibility in OCD with the hypothesis that it is severely impaired. And indeed, that's the case. This is the so-called CANTAB test, which we invented to test cognitive flexibility, where the subject on the touch-sensitive screen responds to these icons to find out which is rewarded. And what you have to focus on is that one of the shapes will be rewarded, and these lines are irrelevant. Even when they're superimposed on the shapes, they're completely irrelevant. Even when you swap the stimuli to make a reversal task so that the subject has to respond to the other shape, lines are still irrelevant. Even with new shapes and lines, shapes are still the main thing to focus on, and lines are irrelevant. But eventually, lines suddenly become relevant. So you have to make this switch from focusing on shapes to responding to lines. And it turns out that patients with OCD and several other mental health disorders are very bad at this. And Mathilde Vaghi showed this rather beautifully. Here are the errors by two groups, controls and OCD patients. You can see at the critical stage, they were completely normal until that stage. At the critical stage, they make many more errors than control subjects who are matched for IQ and age and also gender. But intriguingly, performance on the test is also correlated with activity in the brain. And it's correlated with a particular pathway in the brain from the frontal lobe, from the ventrolateral frontal lobe areas, which we already suspected because of some experiments we'd done earlier in humans and other animals, projecting to a subcortical structure called the caudate nucleus. And the activity in this pathway predicted the performance on this test of extra-dimensional set shifting or cognitive flexibility. So we think that these OCD patients have weakened activity in this particular network in the brain. And that's part of a much bigger story, which I don't have time to go into, but it illustrates certainly some of the functions of the frontal lobes. Now, another major disorder, of course, is attention deficit hyperactivity disorder, which occurs in juveniles, adolescents, but also in adults, actually increasingly. And these disorders also probably have a somewhat genetic basis in this case, and also a brain basis. There are some evidence for this, associated with some problems in the dopamine and also the noradrenaline transmitter systems. And this can be related to some of the specific symptoms, again, by using particular tests in the laboratory. And one of these tests is the test of inhibition, because patients with ADHD are often very impulsive and find it difficult to control their behavioral output. Well, this test in the lab tests this brilliantly. So the computer projects these arrows to the right or to the left rather quickly, and the subject has to respond to the left or to the right on two buttons. But on some trials, a beep sounds, and the auditory beep says, "Don't respond on this trial." And the beep might be sounding just as you're pressing the button, so you have to inhibit this response which has been initiated. And your ability to do that can be measured by mathematical modeling of your reaction times to derive a measure called the stop signal reaction time. And the longer that is, the more impulsive you are. It takes you longer to inhibit a response. And as you can see here, patients with ADHD are very slow on the stop signal reaction time test. They're bad at inhibition compared to controls. And drugs such as Ritalin or methylphenidate, or a drug affecting noradrenaline, atomoxetine, or Strattera, as it's called in the market, are good, in the laboratory at least, at improving performance on this test of impulsivity, response inhibition. Now, this test also is sensitive to frontal lobe function, to a rather different bit of the brain than we've been discussing earlier for working memory, which was here. We tested in this series of brain damage patients a large number who had damage which extended over all the major sectors of the frontal lobe, which we've marked in red. And then we worked out the volume of damage in each of these areas and did a multivariate analysis to relate it to behavioral performance. And the area which comes out as most faithfully reflecting the stop signal reaction time rather well here is this inferior frontal cortex region on the right side of the brain. This area is very much involved in response inhibition or braking. And we've shown this also in brain scanning experiments. So these are, again, brain scanning during the stop task, where they're treated with this drug atomoxetine, which we know is useful in ADHD. These are healthy volunteers, I should say. Now, atomoxetine improves performance even in healthy volunteers, and it also enhances the activation of this right inferior frontal cortex region here. So we believe that by modulating this network involved in the right inferior frontal cortex with noradrenaline in this case, this achieves a remediation of this impulsive tendency. Okay, so also stop signal inhibition is related to other disorders. So Karen Urscher and I have studied patients with stimulant drug addiction, that's addiction to drugs such as amphetamine, and found intriguingly that they're very bad indeed at the stop task. They can't inhibit their responses. But intriguingly, nor can their relatives, their first degree relatives, who are not addicted to drugs. So we think this may run in families in part. And we also found that this is related again to a loss of white matter, connections in other words, again in this area of the right frontal lobe. So this may be a predisposing factor possibly for addiction, this ability to control your impulses, in this case, to control your impulses to take drugs. Okay, so in the last part of the lecture, I want to come on and talk a bit more about hot cognition. So I've mainly been talking about rather cold abstract cognition, like playing chess, for example. And there is this part of the frontal lobes, the orbitofrontal cortex, damaged, remember, in Phineas Gage's case. This part of the orbitofrontal cortex is connected subcortically to the reward system of the brain, which is modulated by dopamine and serotonin. And this is the red hot loop here, which is the schematic of the actual loop in the human brain there. We've already studied some of these other loops, in fact, mediating other forms of cognitive performance. This hot loop is especially interested in emotion and motivational processing. And Damasio, Antonio Damasio, a neurologist working out of Iowa City, discovered or rediscovered Phineas Gage, and also in relation to some of his own patients who'd suffered damage to the same area of the frontal cortex as had Phineas Gage, often due to surgery for tumors, for example, or strokes or whatever. And this damage, as you can see from these sections, is very much in this orbitofrontal ventromedial area. You can see very clearly, I hope, here. And one of his most famous cases, EVR, was a chief accountant and a natural leader with superior IQ. But after his meningioma resection, he suffered personality changes. He was perfectly good at tests of working memory and the Wisconsin card sort test and tests of cold cognition. But what he was terrible at was decisions in everyday life. He made poor business decisions. He became bankrupt. He lost his job. He got fired from others. He was divorced. He remarried a hooker and divorced after 2 years. And he also showed OCD symptoms and hoarding behaviors. But he was completely normal in many aspects of cognition. So he's like a modern day Phineas Gage. And one of the tests he was bad at in the lab was the so-called Iowa gambling task. So in this test, you have to pick a card and try and win money from one of 4 decks. These two decks give really big rewards, but horrendous punishments occasionally. These other decks, C and D, are steady earners, small rewards, hardly any punishment. Most normal subjects learn eventually to avoid picking these decks because you become bankrupt. And you can see this learning in the normal case. But patients with damage to the orbitofrontal cortex, as you see here, do not learn this. They continue to take enormous risks and inevitably become bankrupt. They also don't form hunches about the behavior. So normal subjects, when they're doing this test, show changes in skin conductance associated with the dangerous cards, even before they're aware that they're dangerous. So they kind of unconsciously move their choices to the safer cards, as shown by this skin conductance response, as you can see here. But patients with damage to the frontal lobes show none of this, as you can see on the right. So these anticipatory skin conductance responses, which differentiate the two decks, are completely absent. So they're unable to link their gut feelings to cognition. This is Damasio's big hypothesis. Now we've got our own test of decision making. It's called the Cambridge Gamble task, and it doesn't involve learning, and it's quite quick and elegant, we think, for testing other facets of decision making. In this test, you have to guess where the yellow token is, and you have to guess so many of your points on this. And if you're right, you win the points, and if you lose, you subtract the points, and you may become bankrupt, but you're urged not to become bankrupt. So that's a win, as you see. Patients with damage to this area, the ventral medial prefrontal cortex, show abnormally high bets compared to patients with dorsolateral damage who have problems with working memory, but not with this particular kind of decision-making task. Now, why I introduced this task is because we studied the effects of the serotonin system modulating performance in this area of the brain. Now, serotonin is another one of these chemical transmitters which originates in the hindbrain and innervates wide areas of brain, in particular this orbitofrontal ventromedial prefrontal area. And you can study serotonin by using drugs. For example, Prozac, the antidepressant, is a drug which boosts the serotonin system. Another way of studying it is to deplete the brain transiently of serotonin with the dietary manipulation, because most of your serotonin comes from tryptophan in the diet. And if you give an artificial diet without tryptophan, even in the human brain, there's a small dip in serotonin, which reversibly recovers, but is enough to be able to test the effects of this treatment. And as you can see here in this huge study where we compared brain damage patients, drug addicts, and people, healthy volunteers with low tryptophan or low serotonin, you can see in every case they perform worse in this test of decision making. So serotonin contributes to this decision making apparatus in the orbitofrontal cortex. And we think it also contributes to other kinds of decisions, such as moral judgment. So here's a famous dilemma scenario where many of you may be familiar with this, where one has to make a choice suddenly between, you're the points operator here, and you have to make a choice as to throw the points to save 5 people at the expense of 1 individual. Would you do that? That's a utilitarian type of dilemma. And an even more upfront one is where one would push someone over a bridge in order to save these 5 people. This is obviously a very personal decision, and generally, intriguingly, quite a difference between these two for individuals. Many individuals who make the utilitarian decision will not make the decision to personally knock someone into the path of the oncoming trolley. So intriguingly, this task is sensitive again to damage to this region of the frontal lobes. And it's also sensitive to serotonin. So Prozac, for example, makes you adopt more personal decisions, makes you even more loath to avoid committing this kind of act. and makes you less utilitarian in this particular example. So serotonin modulates this decision-making capacity, even in this very sophisticated form of decision-making. And indeed, part of this frontal lobes is called what's known as the default mode network from brain imaging studies. And I don't have time to go into this in detail, but you can see here, again, these are images of brain scanned activities in orange in this case. And the medial parts of the frontal lobe, this again, the medial surface, are activated by tests not like the Wisconsin card sort test or working memory, but more tests which are about thinking about oneself, reflecting on oneself, or reflecting what others think of you, and making indeed moral judgments. So this part of the brain is very much involved in reflection about the self and internal decisions, if you like. And then finally, perhaps I can bring this together in quite raised by our former Foreign Secretary, David Owen. As you know, the youngest Foreign Secretary in the 70s, a neurologist by training, went out of power eventually, but has written some very intriguing books about the power of disease and also power itself to corrupt political decision-making. And David has suggested a new psychiatric almost classification called the hubris syndrome, which is observed over many years, occurs particularly in politicians at the very zenith of their career. So he describes this as an acquired personality disorder, and his evidence is based on a study of US presidents and UK prime ministers over the last 100 years. So you're very welcome to think of your own candidates who would take part in this hubris syndrome classification. But he describes this in this very nice paper in Brain, 2009, as well as the book, The Hubris Syndrome. Now, what is the hubris syndrome? Well, let's look at some of the symptoms. A narcissistic propensity to see one's world primarily as an arena in which to exercise power and seek glory. This is all about the default mode network. A disproportionate concern with self, a messianic manner, excessive confidence, exaggerated self-belief, accountable solely to history or God, a loss of contact with reality, often associated with progressive isolation and taking advice only from a very small body of evidence and maybe only one or two advisors if you're a politician, and ignoring all the rest of the evidence. And coupled with recklessness, impulsiveness and inflexibility. So these are the ingredients of the hubris syndrome, many of which you'll have noted have occurred following frontal lobe damage or dysfunction. And we've got a hypothesis about the hubris syndrome that indeed it is related to frontal lobe dysfunction. And it's also related to the subcortical transmitters I've been talking about, which mediate the effects of stress. So the basic idea is that people in these very elevated positions are under almost constant stress. And eventually the stress produces changes in these transmitter systems where demand overtakes supply of the transmitter, and the ventral medial prefrontal cortex becomes depleted of these neurotransmitters. So there's plenty of evidence to support that idea, mainly from animal but also human studies. And in particular, there's some very interesting evidence which is supposed to show that people in high positions do better than people lower down. For example, in the civil service, that's been claimed by Marmot. However, at the very highest levels of the hierarchy, life at the top, for example, in government, may involve a different kind of neurobiology, as has been shown in the baboon colonies, where the very top alpha baboon has been shown to actually have increased stress, increased glucocorticoid levels as an index of stress. So the really top alpha baboon people are constantly worrying about whether they're going to be displaced and are under constant stress. And we hypothesize eventually will make disastrous decisions. So therefore, the hubris syndrome could be an inevitable consequence of risky decision making under stress at the top of the human social hierarchy. So that's our hypothesis, if you like. So in conclusion, I think it's time to stop. It's 10 to 7. The frontal lobes, what I've been talking about is only connect in the Forster sense, emotional and rational thinking. The emotional fuel coming from some of these subcortical transmitter systems, in particular directed to the orbitofrontal cortex, that ventral medial part of the frontal lobes, and also affecting the cold cognition involving the dorsolateral prefrontal cortex and the ventrolateral areas on the right side and the stopping system. And these areas together working as a kind of executive committee to control your behavior, rather than a little man in the head who makes decisions. Thank you very much.
Thank you very much, Trevor. Thank you. And thank you to Ella for removing the slides. That was absolutely fabulous. I was completely engulfed there. And thank you for bringing it around so topically at the end as well. I've got a whole host of questions. So we've got just 10 minutes. So if we just run through some of these. So there's one here from Nicola, who asks, do patients who have anomia have mainly damage to the frontal lobes or other areas of the brain? Trevor.
Anomia. Well, anomia is a problem.
Sorry.
Is that anomia?
Yes, anomia. So word finding.
Word finding, yeah. Yeah, word finding is a problem often associated with damage to the temporal lobes of the brain, back here, rather than frontal lobes.
Yeah.
So frontal lobes is involved in verbal fluency. So if I set you a test to name as many words beginning with the letter F as possible in 90 seconds, that's a test of letter fluency. That would really stress your frontal lobes, particularly on the left side in Broca's area. But anomia per se is very much associated, I think, also with temporal lobe damage.
Okay, thank you very much. So from Annabelle Curl, has the neuroinflammatory environment been explored in OCD and ADHD? Example, microglia activation, that could happen.
Increasingly, yes, it has. I mean, that's a point of big interest in Cambridge, because Ed Bullmore has written this lovely book on the inflammatory response called the something like the inflamed brain or something like that, which is talking about this relationship between inflammation and brain function. For example, in patients with arthritis. And indeed, there's been a lot of work on schizophrenia, in particular in depression. I think depression is one of the better examples. There is a major paper in OCD by Jerome Mayer in Canada, which has indeed shown some microglial changes in adult patients with OCD. And of course, you're probably aware of the phenomenon of pandas, which is, it's a bacterial infection that you get in childhood, which damages part of the circuitry involved in OCD, which includes the caudate nucleus. So there's certainly big links between inflammation and frontal lobe function.
And can I just follow on from that? Is that then inflammation as a form of damage, or is it specific to the kind of damage?
Well, I think that's the big question, actually.
Yeah. Okay. Sorry.
Yeah.
So yes, here's a big one, philosophical one from Freddie. What's required to be conscious?
It's very interesting. Well, it doesn't do to talk about conscious until you're very old in the tooth as a neuroscientist, and I'm not quite old enough, although I'm pretty old. So I don't like talking about consciousness. However, I will say a few things. I think it's multifaceted. So obviously, one very big aspect of consciousness is coma. And, those poor patients in vegetative state, I mean, they're clearly unconscious. That's a very basic arousal function of the brain. That's rather different from the phenomenon of blind sight, where you have damage to the visual system and you're apparently blind, and yet unconsciously you can feel your way around. So you must have some residual visual processing going on, which you're unconscious about. So that's a very intriguing aspect. And I think working memory has been linked to consciousness as well, because people think of it as like a global workspace. It's like your desktop. You know, working memory is what's going on at the moment, and you're manipulating things, and that's consciousness. You know, I think consciousness, importantly, is promoted by some of the ascending systems from the brainstem arousal systems, like noradrenaline, for example, and acetylcholine in particular. I think without those systems, your consciousness fluctuates a good deal.
Thank you. I was thinking when you were speaking that obviously we study pathology or disorders in order to understand the way the brain or whatever system we're studying really works. So looking at the disease state to tell us, what's normal, how do we work? And but with this work, is there also, but does it also lead, I should know this, but obviously does it also lead to then improved treatments for the people who are you know, particularly in instances of brain damage and so on, stroke?
Well, I think so. I think one example, to be honest with you, a lot of drug treatments in psychiatry have worked by serendipity. You know, people tried things and they worked. They didn't have a really good theory for it. And we're still trying to catch up on that, to work out how these things actually work to produce benefit. And I showed you one example there with atomoxetine in ADHD, how atomoxetine may actually improve impulsive behavior by simulating this in a lab experiment in the brain scanner. So noradrenaline was boosting this frontostriatal system. So that's one example. There are many other examples. I mean, very interesting treatments for OCD, very severe OCD, include something called deep brain stimulation. So these are for patients who are spending 16 hours a day doing their rituals. Their lives have disappeared and they beg, you know, some treatment. So the neurosurgeons will implant electrodes, quite tiny electrodes actually, which can be stimulated and which will change transmission in particular circuits. And I've recently been involved in one of these experiments, and we've shown that 2 of the circuits which have been implicated in OCD, if they have electrodes implanted in them and modulated, achieve absolutely amazing effects.
Thank you. Some more questions. So from Mary, would you recommend that older people should take neuroenhancement drugs to boost memory and cognitive function?
Well, I mean, if they've got Alzheimer's disease, they probably should try that.
Yes.
You know, the cholinergic drugs like donepezil do have some effect to slightly improve performance for a while, you know. So it is theoretically feasible, but I don't think I'd necessarily recommend that normal elderly take memory enhancing drugs, although you know, there are some drugs which are relatively safe, and you know, if you think they help you, maybe you should try them. I don't know. Modafinil is a very good example. Modafinil is an artificial stimulant drug which produce small boosts of cognitive performance in certain sectors. Doesn't seem to be an addictive drug at all. Has been licensed in the treatment of narcolepsy. It stops you from falling asleep. And it has mild cognitive enhancing effects. Now, whether or not these would work in real life as disposed as to the laboratory, you know, we're not sure. But many students are supposed to be taking these drugs, so somebody must think they will.
Don't encourage them.
I certainly wouldn't.
No. I think there are more questions coming on and lots of thank yous, Trevor, but I think in the interests of time, because we're coming to an end here, I think I'll just say, you know, a massive thank you, because I hope my medical students are present. I'm hoping that Ella will tell me I can see who's been here at the end of this. Because you know that was just wonderful. It was wonderful to go over all the different roles that the frontal lobes play, and just that connectivity between the different parts of the brain, between the different cells, and actually really visually, which was a massive help for me.
It's an absolute pleasure. If anyone wants to email me on TWR2, with a burn TWR2 at CAM,
Yep.
etc. I'm very happy to answer questions. Okay.
Thank you so much. And you can expect some questions, I suspect. Absolutely wonderful. Thank you so much. And I don't know, it'll have to be a virtual round of applause, I'm afraid. You can use your little hand function, but it feels ridiculous if I do it because it sounds really daft. But thank you so much. And there's loads of thank yous there in the chat function. It just remains to say good night and thank you to everybody for attending and taking part and asking questions. Much appreciated.
Good night.
Good night.